Hi, this is Neela Vakore, and in the previous class we discussed about the cell, plasma membrane. We are going to continue with the same, that is plasma membrane. So here we are going to be talking about the special features of plasma membrane. We have seen the three models which were given by three groups of scientists. The first one was sandwich model, then unit membrane model, and the most accepted one, which is the fluid mosaic model. So keeping in mind the model which has been accepted, that is fluid mosaic model, we are talking about the special features of plasma membrane. The most important thing is that it is made up of proteins and phospholipids. Then it is a trilamellar structure. It has two types of proteins. The proteins are of two types, the extrinsic and intrinsic. Let us take one example of each. Example of extrinsic can be taken as ATPase, and of intrinsic, cytochrome oxidase. These proteins are found as enzymes or carriers during electron transport chain. Extrinsic proteins make about 30% approximately of the complete protein component of plasma membrane, whereas intrinsic proteins make about 70% of the total component. So 30% and 70. Now this is approximate. In different areas, the protein extrinsic intrinsic ratio may change. Fourth thing, the phospholipids. Now let us go back to that structure which we made in the previous section. We made, and we are talking of the fluid mosaic model. I am drawing this so that we are able to recollect what we did. These are the extrinsic proteins. These molecules are phospholipid molecules, and this protein is an integral protein which is actually through and through. So we can also call it transmembrane protein. Now let us come to this phospholipid. So we are talking about these phospholipid molecules which are present in the plasma membrane. If this is the outer side, this side becomes the inner side, and here would be cytoplasm and everything else inside the cell. So amongst the phospholipids which are predominantly found in the outer layer of phospholipids. So outer layer has predominantly two very important phospholipids, that is cephalin and sphingomyelin. These are two which are found predominantly in the outer part, that is the outer layer of phospholipids. In the inner layer, again there are two phospholipids which are predominantly found. One is lecithin and other is phosphatidylserine. So there are two predominant phospholipids which are found in the outer layer, and two, that is lecithin and phosphatidylserine, They are found in the inner layer of phospholipids. Another important thing, plasma membrane also has cholesterol. This cholesterol is present in between the phospholipid molecules. So these structures which we have drawn here, these are cholesterol molecules. What is the role of cholesterol in plasma membrane? It gives strength to the plasma membrane. In prokaryotes, cholesterol is absent. So instead of cholesterol, there is another substance which is pentacyclic sterol. So in eukaryotic plasma membrane, cholesterol is present. In prokaryotes, because it's only the outer membrane, there is no inner membrane present, but that membrane would have another thing other than cholesterol. It is pentacyclic sterol. Another important characteristic feature of plasma membrane is that it has about 20 to 25% water. These are some important things apart from what we have already done, that is the thickness of plasma membrane. It varies from 70 to 100 angstroms, and plasma membrane is porous, it has pores, so the pore size is 7 to 10 angstroms. So these things we have done when we were doing the structure of plasma membrane. So these are some very special features of plasma membrane. Now we were talking about modifications of plasma membrane. There are certain areas, certain places where plasma membrane shows certain different things. We call it modifications. Once we take examples, it will be clear to you what exactly we are talking about. The first one, microvilli. We have heard about this microvilli. These are small finger-like structures which are present in certain cells. So if this is a cell, the upper free surface would have these finger-like structures which are called the microvilli. These microvilli are formed because of the foldings of plasma membrane. The size of these microvilli is 0.6 to 0.8 microns, and their number can be up to 3000 per intestinal cell. In intestine, these cells which are present, they have these microvilli. Each cell can have up to 3000 microvilli. The function of this microvilli increase the surface area. And why should the surface area be more? It should be more for absorption. So this is one modification of plasma membrane. The second modification of plasma membrane is, actually these are multiple, we are talking about it as junctional complexes. Junctional complexes. Let us take different types of junctions. As the name tells us, junction means these are between two cells. First are called interdigitations. We know digits means finger. So interdigitations are some folds. Say this is one cell, say cell A, and its one side of plasma membrane is folded in the form of some finger-like structures, and this is the other cell, and its plasma membrane is also folded like finger-like structures. The only thing is these digits, they fit into each other like this. So now, if it was a simple cell membrane like of cell A and this of cell B, how much area would have been available for exchange? Only this much. But if there are folds, then the area for exchange of substances between cell A and B or two adjacent cell becomes more. So this is interdigitations. Second are known as tight junctions. Tight junctions are formed. Again, this is a junction between two adjacent cells. The two cells, and just to understand this structure, I am making this phospholipid layer and the proteins. This is one plasma membrane, and say let us see, this is the other phospholipid layer of the other plasma membrane of the neighboring cell, and these are the proteins of the other plasma membrane. So this is the plasma membrane of cell A and this is of cell B. Now in tight junctions what happens is the proteins of one layer and the proteins of the next plasma membrane, they fuse, and these are known as tight junctions. There is a technical name given to this tight junctions. They are known as zonula aculodense. This term is very, very important because it's a technical term. Commonly we call it tight junction. The third modification, we are talking about again junctional things. So this is interdigitation, tight junction. We are taking the third one. It is known as desmosome. In desmosome, the gap between two plasma membranes, again, this is one, this is another one. If it is a normal thing, there would be a gap which is known as intercellular gap. But in case of desmosomes, these gaps, they increase. So now it appears little wider at these points. So now there is a wider area between two plasma membranes. These two adjacent cells, they secrete some cement-like material here, and from these materials inside the cell extend some fibers. These fibers are known as tonofibrils. So it is sort of a binding, so you can say a welding type of thing which is happening between two cells. This is A cell and this is B cell. So the intercellular space or gap widens, a cement-like material or a protein again is secreted by both the cells, and in this particular area there are fibers which are extending into the cell. Technical name given to desmosomes is macula adherens. Adherens, adhesion is to stick. So they are helping these two cells to stick to each other. A slight modification of desmosome are known as terminal bars. So terminal bar becomes another modification of plasma membrane under junctional complex. Everything between desmosome and terminal bar is same except that these tonofibrils are not there. So again this intercellular space widens. There is a cement-like substance which is secreted. This is cell A, this is cell B, and here we are not seeing the tonofibrils. So the difference between these two things is only one. Technical name given to terminal bars is zonula adherens. Both these are adherents. This is also for adhesion, this is also for adhesion. That means these two things are acting as welding things, holding both the cells together. Difference is, in desmosomes we have tonofibrils, whereas in case of terminal bars, these tonofibrils are not there. Let us take one more modification under junctional complex. These are known as gap junctions. Gap junction, scientific name given to it is nexus. What happens in nexus is, between two adjacent cells there is a septum formed. So one plasma membrane and another plasma membrane, and there is a bridge which is formed. So there is a bridge between two cells, and this is known as gap junction. One more type of modification is seen under this. Let us write it here. These are known as plasmodesmata. What happens in plasmodesmata is, a similar kind of bridge between cell A and cell B is formed. So this is cell A and the plasma membrane of cell B. There is a cross bridge which is formed. Plus, in this cross bridge, the cytoplasm which is here and cytoplasm which is of cell B, they are connected by cytoplasmic strands. So through these gaps which are found in gap junction, gaps are here also, but there are cytoplasmic strands which are present in between the two cells. Function of plasmodesmata is exchange of substances which can take place between these two cells. So these modifications of plasma membrane come under junctional complexes. We have talked of 6 interdigitations, then tight junctions, desmosomes, terminal bars, gap junctions, and plasmodesmata. Now let us take one more modification of plasma membrane. So first was microvilli, then was junctional complexes. Let us talk about the next one. The next modification of plasma membrane one is sheath. Sheath is present in cilia and flagella. Normally when we see a cilium or a flagellum, we draw it like a thread-like thing coming out of a cell. But if we actually magnify it, what we see is, this is the plasma membrane. The cilium or flagellum arises from a basal granule, and this is the structure, cilium or flagellum. If you observe closely, this plasma membrane is extended on the base of the cilium or flagellum. This extension is known as sheath. This is just to provide additional support to cilia or flagella. Another modification is formation of endocytotic vesicles. Endocytosis is a process when the cells take in some bigger molecules. We have done this earlier in junior classes about phagocytosis, like how amoeba engulfs the food. So if this is the plasma membrane and it encounters the food particle, this is plasma membrane, what happens is the plasma membrane shows a cup-like thing, cup-like structure formed, and the food is taken in. After some time, the complete food particle is engulfed. So this is the endocytotic vesicle which is formed. This structure which is formed is endocytotic vesicle. This is also a modification of plasma membrane that we see. One more endocytotic vesicle is formed, which is called receptor mediated endocytosis. This complete process is also explained with the help of a simple abbreviation RME. It is receptor mediated endocytosis. What happens in this case is same plasma membrane, but the plasma membrane has some receptors. So there are receptors on the plasma membrane. Now, sir, if certain specific molecules are to be taken in, those specific molecules, for example these, they will bind to these receptors. So now this plasma membrane with receptors shows a depression. It is known as a pit. So it forms a pit, and in this pit are these receptors now. These special molecules, they bind to these receptors, and this plasma membrane further becomes cup-like. So again the receptors are in here, and attached to these receptors are those special molecules which are to be taken in, and in the last stage what we see is the complete area with the receptors has gone in, and attached to each receptor are these specific molecules which have been taken in now. So the difference between the two things is this is also endocytosis, this is also endocytosis. In this case, the plasma membrane forms only a cup-like depression. The food particle is taken into this cup-like area and it closes and then goes in. In receptor mediated, again endocytosis only is taking place, but it is mediated or helped by the receptors. So the plasma membrane has receptors. Attached to these receptors there are molecules which are taken in, and again a cup-like thing is taking place. So endocytosis can be of two types. What happens in exocytosis? The reverse process. So in reverse process what will happen is, it is known as exocytosis or ephagy. In this case, the plasma membrane from inside, the vesicle which is having undigested food comes closer and it opens here. So when it opens, all that material is excreted out, and this membrane of the vesicle becomes the part of the plasma membrane. So this is a reverse process. In both the cases, plasma membrane is undergoing some modifications. So formation of endocytotic vesicle is one more modification of plasma membrane. Another modification of plasma membrane is known as cell coat. Up till now we have talked of plasma membrane being made up of proteins, phospholipids and proteins. In this case, the outer proteins, here are these phospholipids, inner layer of phospholipids and then inner proteins. In this case, we have only the outer layer which is taking part into this cell coat formation. Now this is protein. There are two possibilities which can happen here. To the proteins can get attached lipids. So this complex becomes a lipid protein complex. What is attached here is a lipid which is attached to the protein. And the other one is a carbohydrate, especially an oligosaccharide. Gets attached to the protein. Now this complex, that is protein and oligosaccharide complex, is known as a glycoprotein complex. So there are two things, glycolipid complexes and glycoprotein complexes. Glycolipid and glycoproteins. Glycolipids make an additional layer over it, which is for protection. And glycoproteins, they help in cell identification mainly. Cell identification. So especially this part is also termed as MHC, major histocompatibility complex. What exactly is meant by this? We have heard of blood matching, tissue matching. Before the blood donation of a person has to receive blood, there is blood matching, blood group matching, which we call. So basically what are we trying to see is that these glycoproteins which are present on RBC of the donor, are they similar to the glycoproteins which are present on the recipient's RBC? If it is not, then the body would reject that particular cell. So this is cell identification, which we can also term as cell matching. So there are two things which are here, glycoproteins and glycolipids. Together they make the cell coat, which is technically also known as glycocalyx. Another modification of plasma membrane, actually not a modification but something which is present in the plasma membrane, are pores. So pore, the openings. Now opening is not actually an opening, there are some proteins which are acting as channel proteins. These pore size is from 7 to 10 angstroms, which we have seen in the case of the structure of plasma membrane. So these are some modifications which are seen in plasma membrane apart from its original structure. In different cells and different structures of the cells, the plasma membrane is the same, that is protein lipid layer, but the ratio of protein and phospholipid may change, plus the thickness of the plasma membrane may also change. So when we are using the term plasma membrane, we are normally referring to the membrane of the cell, this membrane which surrounds the cell. What about the membranes which are of the organelle? That means the membrane of mitochondria or membrane of nucleus or membrane of Golgi body or anything. They are pretty much same except for the thickness and the composition of proteins and phospholipids. So when you are talking of all the membranes together, we normally use a term called biomembranes. Now biomembranes would complete every kind of membrane. What is the main role of this plasma membrane? Plasma membrane is a physical barrier between, if this is a cell and this is the plasma membrane, it is a barrier between the external and the internal environment. It separates the cytoplasm from the surrounding or from the outer environment. Plus, it also helps in exchange of substances. Plasma membrane plays a very important role in exchange of substances other than these some modifications which we have seen. So now let us take transport of substances across plasma membrane.